Okay. Meanwhile, others are coming. You can ask me if you have any query from the previous two lectures. If you want to ask anything which you have not understand or any other questions you are having. So anyone can ask. You can unmute yourself and you can ask me whatever query you are having. Does anyone is having any query?
Sir, yesterday I was asking about ribozyme.
Okay, yes. Tell me. Ask your question.
Sir, how the ribozyme is used in antisense technology? Because I answered antisense technology, or you didn't respond this.
Okay, fine. So what I'll do, I'm ribozyme. Actually, we are going to talk in detail the different usage of RNA. So when we will talk about the transcription process, that time we will see what are the different usage of RNA, where we are going to use it, and then we will talk about different usage of ribozymes in the regulations, also in kind of other activities, also as enzymes also. So there I will tell you the details. Just a few lectures, after few lectures. Will it be fine?
Okay, sir. Thank you, sir.
Because at the same time we will talk about spliceosome also. So there I will tell you the detail. Okay.
Okay, sir.
Fine. Anyone else is having any issue? Any questions? Okay. Shall we start?
Yes, sir.
Okay. I'm coming to this DNA replication part. So what I'm going to do today is, so first, yesterday someone has asked about the karyotyping or some questions. So something I have told them, and I told them that I will show you the picture. So I'm going to show you the karyotypic picture, and how we are going to study the chromosomes, and then we'll come to the DNA replication portions. There we will talk about the different types of modification in the histones, and how those modifications is going to affect euchromatin and heterochromatin interconversion. And then we'll come to the replication portions, replication part. So this is our chromosome. Probably everyone might have seen this picture. This is the normal chromosome as we are going to see it. But in this chromosomes, it is not the same. Everything same. We are having two chromatids. This is one chromatid and this is another chromatid. So both the chromatids are coming from different parents. So chromatid is one copy of a newly copied chromosome, and they are joined at the centromere portion. So these centromeres are located at different places. Like you can see, if you'll see this central chromosomes, their centromere is in the center. So this portion is exactly same as this portion. Whereas in this case, this portion is not in this, I mean, centromere is not in the center, it is slightly moved upward. So one portion is a little bit larger, another portion is smaller. So one is long arm, other one is short arm. Similarly, here also we are having different types of variations. So normally when we are going to study the chromosomes, we are going to have a shorter arm or a longer arm, most of the cases. So the shorter arms we are going to call P arm. This is short arm, so this is P arm, and the longer arm we are going to call it Q arm. And terminal places we are having telomere, telomerase enzyme, telomeres, which is normally reverse transcriptase type of enzymes. And these are homologous sister chromatids. So roughly, probably when you are going to study about some proteins, you might have seen this type of nomenclature. So on the genes, we are having different types of banding pattern, heterochromatin and euchromatin, or the different type of banding patterns. Those banding patterns are different type of nomenclature. Like chromosomes are divided into chromatids. Chromatids are divided into long arm and short arm. Like this is P arm, short arm. This is long arm, Q arm. Arms are divided into the different regions. Regions are divided into bands or the sub-regions. Sub bands divided into sub bands. So these are different sequentially further division. So like if you are going to see this type of genes or this type of location of the chromosomes, this means I'm talking about the chromosome number 6. I'm talking about the P arm. ER means short arms. There we are talking about the region number 2, band number 1, then subband 3, and subband 4. So this means this is the, I'm talking about one specific gene or one specific location of the chromosome. So every gene having this type of nomenclature. So this chromosome is having many genes, many kind of protein it will give. So every genes are normally named as in these forms. So this is cystic fibrosis. Everyone might have heard about it. Cystic fibrosis is caused by this CFTR gene mutations. What is that gene mutation that is present on the seventh chromosome, and that is the chloride channel. Cystic fibrosis CFTR gene is a chloride channel. So because of that one, normally in our body, chloride and water are interchanged between the inside the cell and outside the cells, and it maintains the, you can call it its jelliness, or you can call it viscosity of the inside the cell and outside the cells. Like this is my cell. So chloride channel is there and water channel is there, aquaporins are there. So chloride can and water can go outside and can come inside. Chloride from chloride channel and water from aquaporine channels. When this gene is going to be mutated, so chloride is not able to cross it, not able to transport it. So what will happen? The viscosity is not going to be maintained. When chloride will not move, water is also not moving. So one side we are having more thick viscous mucus forms, and that blocks the position. Like if CFTR gene is going to occur in the pancreatic duct, so there blockage of pancreatic duct can occur. It can occur in our respiratory tract. It can occur in different other places also. So this is the location of the CFTR gene. Seventh chromosome, Q arm, then region, one band, and this is subband. So this is how chromosome is going to be nomenclatured. Other than this one, there are three different patterns by which chromosome is going to be identified, detected. It will be size of the chromosome, centromere position, and banding pattern. Yesterday you were talking about this one only. Someone was talking about. So you can see in this picture, on the basis of centromere, chromosomes are divided into different kind of names we are having. We are going to have, if centromere is in the center, so this P arm and Q arm will be equally divided. It is called as metacentric, means centromere is in the center middle. If it is slightly moved one side, it is called as submetacentric. This is acrocentric, which is almost near to the terminus end, not exactly terminal, but outside almost the terminal. And in telocentric, it is at the terminal. So one arm is zero, null, no arms are there. Only one Q arm will be there. Then we are having subtelocentric also. So these are different type of, and these are the chromosomes. Like in our case, all the chromosomes are falling in this three category only: metacentric, submetacentric, and acrocentric. We don't have any telocentric chromosomes in our cells. And chromosome number 1, chromosome number 3, 16, 19, 20 are metacentric. Maximum chromosomes we are having, that is submetacentric, that is chromosome number 2, 4, 5, 6, 7, 8. Probably no need to remember, but this is important because sometimes questions comes that which chromosome is telocentric in our case. So we don't have any telocentric, we don't have any subtelocentric chromosomes. X chromosome is submetacentric and Y chromosome is acrocentric. How this one you can see better this picture. So like see, this is chromosome number 1. So if you will see, this is the centromere position. So in this case, chromosome number 1, roughly it is equally dividing the chromosomes. So it is metacentric. Second, I will go submetacentric, chromosome number 2 or 4. So there you can see this centromere is not exactly into the center. It is slightly one side shifted. So it is unequal division of the arms. So this is called as submetacentric. Like in this case, you can see this is almost at the terminus places. Okay. In this case, it is almost at the terminal places. In this case, it is almost at the terminal places. So we are going to study on these chromosomes on the basis of their size. Like if you will start from chromosome number 1, slowly their size is going to reduce. So this is just the arrangement of chromosome on the basis of their size. So that is called as karyotype of human. So chromosome number 1 is the largest chromosome, and chromosome number 22 is the smallest chromosome. Second pattern is on the basis of their centromere positions, which I have told you, like this one. And third is on the basis of banding patterns, like which bands you are going to see it. So this is how chromosome is going to be studied in our case. If you might have heard about, in this case, normally this 21 should be pair only. This is one of the disease, trisomy disease, where chromosome number increased by one number. So is it clear for? I don't know who remember your names, who was asking the questions. Is it clear? Hello. Fine. Okay. Now come to the our actual topic, that is replication. So first, just some I'm going to summarize the nucleosomes chromatin structures. So what we have done till now is our DNA is composed of DNA portion and the protein portions. We are having two types of proteins. They are histones and they are non-histones. Histones have many functions, but when we are talking about the chromosomes, it is in the used in the arrangement of chromosomes. So, in the form of nucleosomes, where DNA will be wrapped on the histone molecule, histone octamers actually, and arrange it so that DNA will be packed inside the nucleus. Other than this one, we are having many non-histone proteins which just supports them, which works as regulator molecules, which works as enzymes, different transcription factors, different kind of polymerases. All those are non-histone proteins. Okay. Then we have talked about sorry heterochromatin and euchromatin. Heterochromatin was the densely packed portion of the DNA, which is normally not actively participating in the transcription and replication process. And euchromatins are loosely packed chromosomes, which normally involve in the transcription and replication process. So euchromatins are transcriptionally active, and heterochromatins are transcriptionally inactive. I have told you how this heterochromatin and euchromatin are going to interchange, how it is going to work. So there are different types of modifications where I, chemical modification which convert euchromatin to heterochromatin and heterochromatin to euchromatin. And this type of modifications normally we are going to call it epigenetic processes or epigenetic modifications. Like you are eating some kind of nutraceuticals and they are going to convert heterochromatin into euchromatin and euchromatin into heterochromatin. So that nutraceutical is going to affect epigenetically. Clear? So epigenetic process means when we are going to do the modification of histones or converting heterochromatin to euchromatin or euchromatin to heterochromatin, it is called as epigenetic process, epigenesis. Okay. In this case, like you can see here, normally this is one kind of modifications, that is methylation. So cytosine is methylated here. So methylation is one process. Other than that one, we are having many different processes. One by one, we are going to see it. This picture you have seen yesterday. Now you can see here. This is, in this case, we are having very compact structure, and this is slightly loosened structure, loosely bond structures. In this case, DNA are not going to be accessed by different enzymes. Like I have to convert into RNA. This DNA needs to go for transcription process. So RNA polymerase needs to bind some places, but DNA is not freely available, accessible for RNA polymerase because it is tightly packed. So in this heterochromatin, where DNA are closely packed chromatins, so there normally transcription process does not occur. So they are going to be repressed. suppressed, inactivated, inactive DNA. When this DNA histones will be modified so that this will be slightly loosened structure so that RNA polymerase can bind to the different portion of DNA and transcription process will occur. So in this case, it is open chromosome structures, why means very easily accessible for different enzymes, different factors, and it is active for transcription. It is activated. It is induced. So it is going to give you different kind of RNA and then further proteins. So heterochromatins are inactive. For transcription, you have to convert heterochromatin into euchromatin. How we are going to convert them? So roughly you remember this. Whenever you are going to have methylation, if histones are methylated, so in case of heterochromatin, methylated histones will be there. So when histones will be highly methylated, it will be heterochromatin. And if you will remove methyl group, heterochromatin converted into euchromatin. If you want, you can write in your notebook that methylation induces compactness in the DNA. Whenever we will have histones will be highly methylated, it means heterochromatin nature compactness will increase and DNA becomes more inactive, transcriptionally suppressed. This is one kind of modification. Second modification is if you are going to add acetyl group, acetylation. So if you will add acetyl group on the histone molecules, so it will be converted into euchromatin. So when highly methylated, heterochromatin nature. When highly acetylated, it is euchromatin nature. Is it clear? Are you getting it?
Yes, sir.
So these are the two major type of modifications which histones gets, and by this they are going to be converted into heterochromatin to euchromatin. But other than this one, there are other modifications also like phosphorylation, ubiquitination. So they also convert heterochromatin to euchromatin. Highly methylation, mainly remember methylation, highly methylation, heterochromatin, and acetylation, phosphorylation, ubiquitinations, then euchromatin. This is broadly chemical modification, but there are some exceptions also there. Means sometime methylation do this one, sometime methylation go this one. It depends on where we are going to do methylation, where methyl group will be added. Like in this case, if I will add methyl group at this place, it will be heterochromatin. If I will add methyl group here, it will be euchromatin. Not exactly this one. I'm going to give you the name of a particular amino acid that you need to remember. So methylations normally gives you heterochromatin, but when you are going to see more detailed structure, that time we come to know that methylations have different type of effect. It that depends on position to position. Like this is the same thing how heterochromatin is converted into euchromatin and euchromatin to heterochromatin. Here you can see it. This is the basic differences. Yesterday also you have seen it. Euchromatins are loosely condensed structure, accessible DNA, and it is active DNA, expressed and active. These are the basic differences between heterochromatin and euchromatin. Whenever DNA will be hypermethylated, hyper means too much methylated, it will be heterochromatin. Whenever DNA will be hypo means very less, then euchromatin. This is broadly. Histones are going to get acetylation. Yes, here I've told you acetylation. When you will do, sorry, here acetylation, it will be converted into euchromatin. But where acetylation occurs? Normally in the H3 and H4. H3, H4 means histone 3 and histone 4s. They are going to get acetylated. So when you will be acetylated your histones, or think about, I have taken certain chemical in my body, I have taken certain medicines in my body, drugs in my body, which do this type of reaction, which is going to do acetylation reaction. So my gene will be active or inactive. I have taken a drug medicine today that is going to do the acetylation reaction of my histones. So drug is going to activate your DNA, activate, because drug will do the acetylation, and when it will be acetylated, euchromatin nature will be activated. It means DNA is going to be transcriptionally active. You have taken certain foods and they are going to increase the methylation reaction, so they are going to inactivate your gene, suppress your gene will not give you proteins. Clear?
Yes, sir.
Okay.
Yes, sir.
Now I have told you for methylation have different effect. If you are going to do the methylation on histone 3, H3 means histone 3, K means lysine, fourth position lysine, because histones are protein. This you need to remember, please. You need to write it and you need to remember. This is your histone protein. On this one, the fourth position, we are having K means lysine. So this is K. If this fourth position lysine is going to be one methylated, only one methyl group, or three methylated, three methyl group will be joined here on this lysine. So if fourth lysine of H3 will be one methylated or three methylated, or lysine at 79th position will be one methylated, it is going to give you euchromatin. Same histone 3, if you are going to methylate at lysine number 19, 9 and 27, it will give you heterochromatin. So lysine methylation at different places gives you different type of effect. If it is position 4, position 79, euchromatin. 9 multiplied by 3, 27, it will be heterochromatin. So it have different type of effect. This is clear?
Yes, sir.
Okay. Now if you want to see little bit more details for acetylation, for addition of acetyl group, we are having a different types of enzymes we are having. Actually, whatever drugs I have told you, whatever nutraceuticals I have told you, whatever food I have told you, they are going to increase acetylation or removal of acetyl group. That is through the some enzymes. What are those enzymes? They are called as HAT and HDAC, histone acetyl transferase or histone acetylase, and removal is called done by histone deacetylase. So if I have taken a medicine which increases this histone acetyl transferase, means more acetylation will be there. More acetylation means gene becomes active. If I have taken certain medicines and they are going to activate deacetylase enzymes, so removal of acetyl group means more inactivation of the DNA. This is the chemical reaction, and this acetyl group is added on the lysine only. So most of the cases lysine is going to be modified. Methylation also, acetylation also, ubiquitination also, phosphorylation also. Most of the chemical modification occurs on the lysine only. So this is lysine molecules at the fourth position. You can call it 79th position, whatever different places. So if histone acetyl transferase will transfer the acetyl group, and this is the acetyl group, COCH3. So when you have this type of a structure, it is heterochromatin. When it will be acetylated, it will be euchromatin. If you want to convert euchromatin to heterochromatin, you need to remove this group. So deacetylase enzyme. Clear?
Yes, sir. Yes, sir.
So any component, any molecule, any signaling compound, signaling system also, which we are going to call transcriptional activator or transcriptional repressor, transcriptional activator normally doing the acetylase activity. They will add acetyl group. It means transcriptionally going to activate your proteins, activate your histone DNA, and those compound which is suppressor, gene suppressing protein synthesis inhibition, they are normally going to do histone deacetylase activity. This is one thing. This is the same thing how it is going to be done, uh, histone acetylase and deacetylase. Similarly, we are having methylation also. We are having methylase and demethylase. So normally if you'll see here, just I will show you this one. This is lysine molecules, and lysine is going to be monomethylated, one methyl group, dimethylated, two methyl group, and trimethylated, three methyl group, which we have seen here in this table. This is one methylation, this is trimethylation, this is dimethylation. Me2 means two methyl group, Me3 means three methyl group. So greater than 90% of methyl carbon is found in the dinucleotide CpG. So here it will be, yesterday also I told you two types of methylation we are going to have. Either nucleotide will be methylated, cytosine will be methylated, or lysine is going to be methylated. So for that one we are having two enzymes, methylase and demethylase. Because whatever we are talking here, most of the study had done in bacteria or prokaryotic system. So methylase, demethylase, deacetylase, acetylase, all these enzymes are present in bacteria. What we are talking about, that is bacterial enzymes. In eukaryotic system, in human, we have similar type of enzymes, but that is not exactly methylase or demethylase. That is, these are the different names we are having. Similar enzymes which do the similar activity, but we are having nomenclature as DNMT. DNA methyl transferase we are going to call in our case, in case of eukaryotic system or human system. It adds methyl group, but it is not exactly called methylase only, it is called as DNA methyl transferase or DNA methylase. So its nomenclature will be DNMT1, DNMT3A, or DNMT3B. So this is how your gene are going to be expressed, transcriptionally active or transcriptionally inactivated. Okay. Other modifications like this methylation I have already seen in that table. So remember this one, K9 or K27 will give you Euchromatin transcriptionally silencing, and modification of K4, K79 is going to activate your transcription. Other than that one, ubiquitination are also there, but that is not that much significant. Methylation, acetylation are two most important transcriptional activator and inducer, which convert your heterochromatin into dichromatin. Now come to the actual replication. Anyone is having any query till now? Heterochromatin, euchromatin, how it is going to be silenced, how gene going to be activated, induced, anything? No one is having any question?
Sir, everything which we eat does the acetylation.
No. Like say, sometime if I am going to eat certain thing and certain medicines, certain food, certain things, it have different activity. Some molecules can activate your gene activation. Some molecules can activate your protein synthesis. Like sometimes we are telling now, if you want to do growth, everyone is knowing, right? Growth means more protein synthesis. So sometime in your family, when you are normally, we are normally kids, that time we used to, our parents used to tell that if you will eat these things, growth will be there, you will grow. It means more protein synthesis will be there, means gene will be going to be activated, then only protein synthesis will occur. So there are certain things which activate your gene, certain things which is going to inactivate your gene. So it will be both way. You have to, means normally questions are not coming on that one, which food is going to activate or which food is going to inactivate. Rather, it have different food will have different activity. Okay. Anyone else? Okay. So now we are going to see the DNA replication. Replication exactly means the copying of the DNA molecules. And remember one thing, when we are talking about transcription process, so first replication, we have transcription and we have translation. Replication, whenever it occurs, the purpose of replication is just duplication of the chromosome. Other than that one, no other purpose is there. Whenever a cell has to divide, then only replication occurs. So if DNA has to go for replication process, it means cell has to divide. Other than that one, no other options are there. No other purpose is there for replication process. So replication only destination is cell has to divide. And second thing is this replication process whenever it will occur for complete DNA, not one specific portion of DNA. So if this was my chromosome, so whenever replication will occur for full complete chromosome from here to here, full DNA. Whereas transcription and translation occurs not for full complete chromosomes, it is for a specific fragment, a specific portion. So only we require certain gene to be expressed at a time. Like in my case, if I have taken my food, my body needs more insulin, so only insulin gene need to be activated. Clear? So replication occurs for complete DNA, transcription occurs only for a specific region of the DNA. And the purpose of replication is duplication of DNA. Cell has to go for cell division. Other than that one, no other purpose, no other aim is there. In our case, roughly we are having 6.5 billion base pair in our DNA of our genome, all of which must be accurately copied. Why we are insisting this word accurately copied is, if you are going to do a small mistake in the copying, it ultimately going to affect my protein. It will leads to mutation, and mutation can give you different type of protein. Sickle cell anemia, how many of you are knowing? How many mutation occurs in sickle cell anemia? One single base pair. So single base pair mutation can convert a uh, normal RBC to a sickle cell RBC, and it will be going to modify RBC and its physiology also. So a small mutation can leads to a means bigger effect also. So that's why we need to copy accurately, and for that one we have different purposes, different things, different kind of checkpoints where we are going to check that it is accurately copied or not accurately copied. The basic mechanism of DNA replications are similar across the organism. So it will be same normally in prokaryotes also, in bacteria also, in fungus also, in human also. There are three different possibility for DNA replication. It can be conservative, it can be semi-conservative, it can be dispersive. Uh, okay. Semi-conservative is, think about, this is the original DNA, parental DNA. I want to copy it. So if I am going to keep this copy intact, no disturbance in this original copy, and exactly same copy, new copy I am going to make it. So this is my original copy which is intact, and the similar fresh copy has been prepared. So in this case, I have conserved my original copy. So this is called as conservative replication process. So this DNA is going for replication in the first generation, so it is intact here, and a new copy has been formed. In the second generation, this is intact, and a fresh copy has been prepared. In for this one, this intact copy has been intact, and a fresh copy has been prepared. So this is called as conservative, where we are going to conserve the parental DNA, precursor DNA. Then we are having semi-conservative. Semi means 50% conserve, half new. So DNA, we know that DNA is double stranded. So one of the strand is from the parental strand from the original from here. One strand this side, one strand this side, and one strand will be newly formed. So this is every time it is going on. So in this case, old strand come here, sorry, and new this strand come here, and a complementary strand will form. Means 50% new, 50% old. So that is called as semi-conservative. And third one is dispersive. Like we have fragmented this whole nucleotides and then mixed together along with the new one and form a mixed portion of the DNA. So all the DNA does not have either semi-conservative or conservative. It is a mixed type of DNA. So that is called as dispersive, a scattered DNA. So these are the possibility. But later on, was an experiment which was conducted by the Messelson and Stahl, and they confirmed that DNA replication is semi-conservative. So our DNA replication occurs not through this process, not through this process. Rather, this is the way by which we replicate our DNA, where one parental DNA is going to be used and another parental DNA will be for another copy, and every DNA will have 50% new, 50% older copy. How to prove that one? For that one, this Michelson has done some experiment. Let me take a blank sheet, and here I'm going to tell you. DNA contains nitrogen that you have seen already. DNA is composed of normally purines and pyrimidines, and purine are having four nitrogen, pyrimidines have two nitrogen. That you have already knowing it. So if your DNA, this is my DNA, if DNA contains nitrogen, so normally we know that nitrogen is having two isotopes, N14 and N15. Which one is heavier? N15 will be heavier, right?
Yes, sir.
It is having higher molecular weight. So when you are going to do the centrifugation, that is CSCL centrifugation, we are doing cesium chloride centrifugation we are going to do. So normally those things which is heavy, they will be settled down here. Heavier things will be in lower side and lighter things will be upper side during the centrifugation process. Okay. This is a bacteria which was normally grown in a culture which contains only one kind of medium, that is N15. So this all the nitrogen of this bacteria is of N15 type. Now if this bacteria is going for replication, so what will happen? One strand of DNA contains N15, another strand here that contains N15, and because our replication is semi-conservative, so a complementary copy will form here. For this generation, this bacteria which has N15 DNA, which is grown in a medium which contains only N14, no N15. So this newly formed DNA is containing N14. So this is N14 and this is N14. I will show you the picture here. Just 1 second here. This is the original DNA. This is from one bacterial cells which is going for replication. This was N15 bacteria which is grown in N14 media. So one DNA is from N15 which contains N15. This is N15, and because bacteria grown in 14 nitrogen 14, so this molecule is half of the DNA is which contains 14. 14 means nitrogen of 14. We have two types of nitrogen in culture. N15 nitrogen, which is the one kind of isotopes, second one kind of isotope, and second one is N14, which is another isotope, which is lighter one. So if you are going to centrifuge this DNA, first one, this one, so in the centrifugation tube it will be near to the base, and if you are going to centrifuge this DNA, it will be not here, it will be in the center, slightly upper side, because some lighter DNA is there, some strong heavier DNA is there. In this case both the strands contains 15 only, so they will be here In this case 50% lighter, 50% heavier, so it will be in the center. And again, for the third generation, again they have grown in N14 media. So only one strands will be 15. This strand will be 15. Remaining all will be 14 only, because bacteria culture is in 14 medium. So this is 14, this is 14, this is 14. So all are 14. In this case, if you'll see, then 50% original type and 50% 14 containing. So in this case, if you will culture it, normally this is the this is my first generation, this generation. So in this generations, only one type of DNA we have got it. They have got only one type of DNA which contains both the strands of 15 type. So their centrifugation will be bands has formed at this location. In the second generations, only one type of DNA. One type of DNA means they contains DNA which is have 50% 14, 50% 15. So slightly above 100% DNA will be of similar type. In case of third this generations, two types of DNA. This is one type and this is another type, mixture type. So two bands has been formed. 50% for this one, 50% for this one. When it next generations, the new DNA numbers has been increased. In this case, you can see 6 new DNA and 2 old DNA. So slightly generation to generation, new DNA length has been number has been increased. So their band has been thickened, and this DNA contribution is only smaller. In case of this one, it is 100% only one type. In this case, 50% this type, 50% this type. In this case, out of 8, 2 of this type and 6 are new type. In this case, out of 16, 14 are of this type and only 2 are of new type. So after this experiment, they have seen that slowly as generation is going on, the contribution, the percentage of N14 DNA is slightly increasing. So if you'll go for further generations, again this line will be slightly more thinner and this will be thicker. Their contribution has been increased. So by this experiment only, Messelson and Stahl had confirmed that DNA replication is semi-conservative. So these are semi-conservative replication process only. This is the first generation where DNA bacteria has grown in N15 media. So only one band has been seen, only one kind of DNA. When this bacteria has been transferred to a medium which contains N14 medium, so bacteria get this N14 nitrogen in their DNA. That time they have two similar type of DNA has been formed. So again, only one band will form in the center between N14 and between N15. 114 and 115 is there. Again, same bacteria has grown in N14 medium. So again, now this band will slightly thickness will be reduced and their thickness will increase slightly. So this confirms that it is semi-conservative mode of replication. That experiment was done by Messelson and Stahl. Clear?
Yes, sir.
For everyone, or shall I repeat it?
Yes, it's clear.
Okay. Now, in our case, if you'll see the bacterial DNA are slightly a smaller DNA and they are circular DNA. In our case, our DNA is little bit longer DNA and it is linear DNA. So DNA replication, whenever it occurs bidirectionally. Bidirectionally means in bacterial case also, if you will see complete DNA, then it will looks like circular. At a time, if you are going to consider only this much fragment of DNA, then it will looks like linear DNA. So in both the cases, in eukaryotes also, in prokaryotes also, DNA replication occurs in both the direction, up this side also, in forward side also, in backward side also. So it is called as bidirectional replication. Tell me one thing, replication where it occurs? Randomly, if this is my DNA, so it will occur at randomly at any place, or there is a specific place from where replication starts, initiates. How replication is going to start?
Origin of replication site, sir.
Origin of replication. Okay. So replication does not occurs randomly. There is a predefined place, predefined location on the DNA strands are there, and from there DNA is going to unwind, because you know that DNA is double stranded, it is helical forms. So you have to unwind the DNA, unwrap the DNA. So both strands need to be separated, and then replication will occurs on both the strands. So that unwinding portion, when it is going to unwrap, you are going to remove this strand to opposite direction. That place is fixed, that is called as ORIC site, origin of replication site. So there are few questions which we are going to say it. Are the parent DNA strands completely unwound before each replication? Like in our case, is it possible that a complete DNA will be unwrapped? Two strands will be separated and then it will replicate together. No, it's not going to happen like that one. There are different location, like replication will initiate at this point, replication will initiate at this point, replication will initiate at different multiple places, and slowly it will move this direction. Here it will replication occur in, you will see later on. So it does not complete DNA does not unwound at a time. It will be multiple small portions is going to replicated, and later on it will be joined together. Second question is, does replication begins at random places or at a unique point? It is not going to occur at random places, rather it is going to start at occurs at a specific place, unique point, that we are going to call origin of replication or ORIC site. After initiation at any point of DNA, does replication proceed in one direction? No, it occurs in both the direction. Like this is one of the example of replication. So if this is the DNA, so think here this DNA has been unwrapped. It is going to form a bubble like a structure. This is called as replication bubble. So here replication occur in this direction also. This DNA will be unwounded and replication will move in this direction. And at opposite end also it is going to unwind in this direction and replication will occur. So replication occurs in both the direction, by direction. Here it is showing unidirectional. So normally we don't have unidirectional replications. Rather we are having bidirectional replication. This complete structure is called as one replication bubble, and half of the portions we are going to call it replication fork. So this is one replication fork, this is another replication fork. When two we are going to mix both the replication fork, we are having a replication bubble, bead like a structure. There are many enzymes, which is many components, many proteins which we did in this process. Just some I'm going to tell you the names, and later on when you will see the functions, how where we are going to use these enzymes, that time then it will you will remember it and you will be understanding. So in bacterial cells we are having three different DNA polymerases. DNA polymerase are those enzymes which in add nucleotides into the DNA and elongate the DNA chain, increase the DNA chain. So you are having DNA chain. Every time one nucleotide will be added there, so that it will be one nucleotide longer. Another nucleotide will be added, and it will be one nucleotide longer. So every time polymerase adds one nucleotides. There are three different DNA polymerase, and all three have different functions, different properties. Normally, DNA polymerase III are the most important enzyme which replicates larger fragment of DNA, larger portion of the DNA. DNA polymerase I, which we normally requires for a smaller synthesis, a small fragment synthesis, a smaller portion of the DNA synthesis. And DNA polymerase II, which normally requires for DNA repair functions. So these are the function of all three. Later on, when we will more compare this 1 and 2 and 3, that time you will see why 3 is used for longer DNA and why 1 is required for a smaller DNA. For time being, for now, it is only this sufficient that 1 is for a smaller portion of the DNA, smaller fragment of DNA. DNA polymerase 3 is larger fragment of DNA. Then we are having helicase enzyme. Helicase enzymes are the enzymes which normally digest the hydrogen bond. because you know that both the strands of DNA are joined together by the hydrogen bond between the nucleotides adenine, thymine, cytosine, guanine. So if you want to separate these two strands, we need to digest this hydrogen bond, and for that one we are having helicase enzyme. So helicase breaks hydrogen bond and unwind the DNA strands. Then we are having ligase. Ligase enzymes normally joins two nucleotides. Uh, this is one nucleotide. This is your chain is going to grow DNA. So when a new nucleotide will come here, it should be added by phosphodiester bond. So ligase enzymes joins different fragment of DNA, nucleotides of DNA by uh, two different nucleotides, one nucleotide here and another nucleotide here by phosphodiester bond. That is ligation. Then we are having primase. Chemically, this is RNA polymerase, which normally makes a RNA portions. DNA polymerase is having one drawback, that it cannot start a fresh copy of DNA synthesis. DNA polymerase can only add a new nucleotide on the growing chain. If you are having pre-existing DNA portion or RNA portion, only their new nucleotide will be added by DNA polymerase. So DNA polymerase cannot start a DNA chain from zero. It needs certain pre-existing fragment. So those pre-existing fragment is going to be produced by primase. RNA polymerase can start from zero. So primase is the first enzyme which start a DNA synthesis, a small RNA molecule actually, and on that one DNA polymerase adds nucleotide. Okay, sliding clamp details, it is normally holding the DNA, and so the DNA strands does not move, and on that one new nucleotide will be added. Topoisomerase, your DNA is like this one, it is coiled structure. When you are going to remove this strand this side, this strand this side, so normally, because of, if in your home also, if you will if you have a rope and you will unwind it, their supercoiling will increase on the further side. So those supercoiling will be removed by topoisomerase or gyrase. Same thing. Topoisomerase and gyrase both are same enzyme, same nomenclature. It removes.
सर ये प्राइमेज वाला एक बार और रिपीट करते
मैं बताता हूँ. See, here. This is my DNA. This is my bubble. I want to start DNA replication from here. This is my DNA, and I want to start from this side. So currently, I don't have second strand here. So DNA polymerase need to synthesize DNA strands, but DNA polymerase cannot add your first nucleotide. DNA polymerase can add nucleotide only on the pre-existing nucleotides. If you are having first nucleotide added, then nucleotide will have 3' OH. On this 3' OH, DNA polymerase can add nucleotide. The first nucleotide need to be there, or some DNA portion should be there, some RNA portion should be there. So DNA polymerase can never start DNA replication from the zero. It needs a fragment of nitrogenous, means nucleotides or oligonucleotide, so that it will add a fresh nucleotide. ये शुरू से जीरो से शुरू नहीं कर सकता, ये सिर्फ न्यूक्लियोटाइड ऐड कर सकता है. आपका पहले से कोई टुकड़ा डीएनए का है, आरएनए का है, या किसी न्यूक्लियोटाइड का है, वहां न्यू न्यूक्लियोटाइड को ऐड कर सकता है, बट जीरो से ऐड नहीं कर, शुरू नहीं कर सकता है. तो जीरो से शुरू नहीं करेगा, तो इसके लिए इसको चाहिए, डीएनए पॉलीमरेज को चाहिए कि पहले से कोई फ्रैगमेंट होना चाहिए यहां. दैट फ्रैगमेंट इज गोइंग टू बी मेड बाय दिस प्राइमेज एंजाइम. So always DNA polymerase needs a primase enzyme or a pre-existing DNA fragment. Clear?
Yes sir, clear sir.
Otherwise, जब हम आगे स्ट्रक्चर देखते हैं ना, how it is going to occur, that time it will be little bit more clear. अब जब आपने DNA, when you have opened this DNA strands, तो बिफोर टिल इट इज गोइंग टू सिंथेसाइज। आपने आरएनए डीएनए में देखा था डिफरेंसेस कि सिंगल स्ट्रैंडेड मॉलिक्यूल्स आर लिटिल बिट मोर फ्रेजाइल, मोर इजीली गोइंग टू बी हाइड्रोलाइज्ड। तो उसको स्टेबलाइज करने के लिए ना वी आर गोइंग टू ऐड डिफरेंट प्रोटीन्स ऑन दिस सिंगल स्ट्रैंडेड डीएनए, सो दैट इज कॉल्ड एज एसएसबी प्रोटीन, single strand binding protein, which bind to the single stranded DNA and then it will stabilize them from the enzymatic activities. So these are the different components which we require during complete replication processes. What are the things we need it? So we require a template. Template means previously existing DNA portions, DNA piece, that is called as template DNA. So you have a DNA strands. In the case of replication, both strands will be separated and both strands will be used as a template. So this is my template DNA. On this one I will add a complementary basis, so it will form a complete new DNA. So we require a template DNA, then require a primer. Primer normal means एक छोटा सा डीएनए का टुकड़ा या आरएनए का टुकड़ा, न्यूक्लियोटाइड का टुकड़ा, जो जहां से डीएनए सिंथेसिस स्टार्ट होगा। हम घर में दही जमाते हैं ना, for that curd we are going to use a starter. So that starter is the primer from where it is going to start, initiate. So this is primers are a small RNA molecules, DNA molecule which is synthesized by primase enzyme. So this primase gives you primer molecule, and then we require DNA polymerase. This is DNA dependent DNA polymerase. Probably आपने देखे नहीं. Total 4 different types of polymerase we are having. DNA dependent. चलिए मैं बताता हूँ पहले, यहाँ देखा. We require a polymerase enzyme. Polymerase means which can add a new nucleotide. This polymerase can be DNA polymerase or it can be RNA polymerase. ठीक है. अगर आपको डीएनए का मॉलिक्यूल बनाना है तो यू रिक्वायर ए डीएनए पॉलीमरेज. इफ यू वांट टू मेक ए आरएनए मॉलिक्यूल्स यू रिक्वायर आरएनए पॉलीमरेज. जो प्रोडक्ट चाहिए वो पॉलीमरेज आपको चाहिए. जैसे हमें डीएनए से डीएनए बनाना है तो हमें डीएनए पॉलीमरेज चाहिए. इफ यू वांट टू मेक ए आरएनए मॉलिक्यूल्स देन वी रिक्वायर आरएनए पॉलीमरेज. अब if you want DNA as a template, so if DNA is my base, DNA is my template, एंड उस टेम्पलेट को यूज करके मैं डीएनए बनाऊंगा, डीएनए से डीएनए बनाऊंगा, तो मुझे चाहिए डीएनए डिपेंडेंट डीएनए पॉलीमरेज. इफ आई विल यूज डीएनए एज ए टेम्पलेट एंड मेक ए फ्रेश कॉपी ऑफ डीएनए, डीएनए से डीएनए बनाना, तो आई रिक्वायर डीएनए डिपेंडेंट डीएनए पॉलीमरेज. If I am going to use RNA as a template and then going to make a DNA, so RNA को यूज करूंगा और DNA बनाऊंगा, then I require RNA dependent DNA polymerase. तो जो आपका टेम्पलेट होगा that dependent DNA polymerase or RNA polymerase. Similarly in this case, if you are going to make RNA to RNA, you require RNA dependent RNA polymerase. If you are going to use RNA and making DNA, so RNA dependent DNA polymerase. So total 4 different types of polymerase we can have, but here DNA polymerase means we are going to make a DNA strands as a template and make a DNA. So DNA to DNA, so we require DNA dependent DNA polymerase. ठीक है ये?
Yes sir. Yes sir.
In this DNA polymerase, there are 3 different type of properties we should have. First one is processivity, second one is fidelity, and third one is proofreading. आप तीनों का टर्म लिखिए अपने कॉपी में, मैं बताता हूं आपको. हमारे घर में नॉर्मली वी आर गोइंग टू यूज ए बैटरी. हमारे पास रिचार्जेबल बैटरी आते हैं, रिप्लेसेबल बैटरी आते हैं विच कैन नॉट बी रिचार्ज, यू हैव टू रिप्लेस इट। आप रिमोट यूज करते हैं जब हम बैटरी यूज करते हैं। आपके पास एक बैटरी ऑपरेटेड कार है या जो भी है, आपने एक बैटरी लगाया, एंड व्हाट इज गोइंग टू हैपन, दिस बैटरी कैन यूज फॉर सर्टेन टाइम, सर्टेन यू कैन कॉल इट साइकिल्स। सो कुछ देर के बाद ये बैटरी डिस्चार्ज हो जाता है, तो यू नीड टू रिचार्ज इट, यू नीड टू रिप्लेस द बैटरी। ठीक है। So similarly for DNA polymerase also, one DNA polymerase cannot have a insufficient, means what we call it, infinity process, infinity न्यूक्लियोटाइड को वो ऐड नहीं कर सकता है। प्रोसेसिविटी मीन्स कितने न्यूक्लियोटाइड को ऐड कर सकता है एक नया पॉलीमरेज। आफ्टर दैट वन पॉलीमरेज बिकम्स inactive and you need to remove that polymerase and you require a new polymerase। एक पॉलीमरेज कितने न्यूक्लियोटाइड्स को ऐड करेगा, उसके बाद आपको नई पॉलीमरेज की जरूरत पड़ेगी। जैसे आपने एक बैटरी यूज किया, कितने देर ये बैटरी चलेगा, जिसके बाद आपको नई बैटरी की जरूरत पड़ेगी। तो पावर ऑफ बैटरी यू कैन कॉल इट। सो प्रोसेसिविटी एग्जैक्टली means number of nucleotide added by a polymerase and after that polymerase need to be replaced, that is called as processivity। कितने देर चल सकता है ये। ठीक है, समझ में आया?
यस सर।
अब आप देखेंगे वी आर हैविंग टोटल 3 डिफरेंट टाइप्स ऑफ डीएनए पॉलीमरेज, 1, 2 एंड 3। लेटर ऑन मैं आपको टेबल दूंगा जहां आपको क्लियर होगा। प्रोसेसिविटी इफ यू विल सी, DNA polymerase 1 can add roughly 3 to 200 nitrogenous nucleotides। Means एक बार आपने ये एंजाइम यूज किया पॉलीमरेज 1, ये 200 न्यूक्लियोटाइड को ऐड कर सकता है, उसके बाद इसकी पावर खत्म, तो आपको नया डीएनए पॉलीमरेज चाहिए होगा। डीएनए पॉलीमरेज 2 कैन ऐड अप टू 1500 न्यूक्लियोटाइड्स एंड डीएनए पॉलीमरेज 3 कैन ऐड more than 5 lakh base pairs, 5 lakh nucleotides। 500,000। हाँ, अब आपको समझ में आ गया होगा कि डीएनए पॉलीमरेज 1 को हम छोटे छोटे डीएनए फ्रैगमेंट के लिए क्यों यूज करते हैं। कि आपको पास मान लीजिए ये 10 लाख बेस पेयर का डीएनए है, अगर मैं ये एंजाइम यूज करूं पॉलीमरेज 1, तो मुझे कितनी बार चाहिए, 1000 के लिए 5, 10,000 के लिए 50, इस तरह से आपको n नंबर ऑफ हजार, ज्यादा मतलब बहुत ज्यादा नंबर ऑफ डीएनए पॉलीमरेज 1 चाहिए होगा 10 लाख बेस पेयर के लिए। जबकि अगर आप ये एंजाइम यूज करेंगे तो ओनली 2 एंजाइम्स आर सफिशिएंट। तो नॉर्मली डीएनए पॉलीमरेज 3 वी यूज इट फॉर लॉन्गर डीएनए स्ट्रेच, डीएनए पॉलीमरेज 1 छोटे छोटे टुकड़े के लिए। सो दैट इज कॉल्ड एज प्रोसेसिविटी। ठीक है। सेकंड we are going to call fidelity, or you can call it polymerization rate। fidelity or polymerization rate means how many nucleotide it is going to add every second। एक स्पीड क्या है, कितना न्यूक्लियोटाइड ऐड करेगा एक सेकेंड में। तो रफली पॉलीमरेज 1 कैन ऐड 16 to 20 न्यूक्लियोटाइड्स पर सेकेंड। Polymerase 2 can add roughly up to 40 nucleotide per second, and polymerase 3 can go up to 250 to 1000 nucleotides per second। So processivity and fidelity both for DNA polymerase 3 is very high। That's why DNA polymerase 3 is normally used for longer DNA fragment, normal means larger DNA pieces for their replications, whereas DNA polymerase 1 is used for a smaller fragment। So processivity and fidelity, that is fidelity means polymerization rate, speed, वो दोनों बेटर है। थर्ड प्रोसेस जो हम बात कर रहे हैं दैट इज प्रूफ रीडिंग। कि आपने कॉपी, आपके पास नोटबुक है, आप नोटबुक में कुछ लिख रहे हैं, मान लीजिए मुझे एक्सो न्यूक्लियेज लिखने हैं, तो मैंने लिखा एक्सो न्यूक्लियेज, यहां मैंने एक स्पेलिंग गलत कर दिया, तो मुझे पता कैसे चलेगा, मैं वापस लौटूंगा, देखूंगा कि वट एवर आई हैव रिटन दैट इज राइट और रॉन्ग. So whatever step you are taking, whatever nucleotide if DNA polymerase adds, it checks that nucleotide added that is correct or wrong nucleotide they have added it. If they have it is correct, then they will move further. Otherwise, if it is wrong nucleotide added, they will remove it and add a correct nucleotide. अपनी गलती को सुधारना उसको बोलते हैं प्रूफ रीडिंग. So they check and remove the wrong nucleotide, add the right nucleotide. That is, so DNA polymerase is having these three properties. We require it. Clear? ठीक है ये?
Yes, sir.
Okay. Roughly, this is the summary of replication. We will go in details. Normally, I am for my study, I am going to use Lehninger book or Harper's book. So if you want, you can read it. If you want soft copy, तो मैं आपको आप मुझे मैं धनंजय सर को दे दूंगा, या मैं उसमें जो टेलीग्राम चैनल में आपने ज्वाइन किया वहां मैंने दिया हुआ है, वहां से भी आप ले सकते हैं. So normally I use Lehninger book. Most of the picture जो मैं आपको later on दूंगा वो Lehninger का ही होगा. ठीक है? सो ये हमारा
सर
यस
क्वेश्चन था
या बोलिए
कि अभी आपने जैसे डीएनए पॉलीमरेज 1 का बताया, तो बाकी जो डीएनए पॉलीमरेज है 2 और 3 उसका क्या है?
नहीं, मैंने तीनों का बताया. यहाँ मैंने कंपेयर तीनों का किया था. प्रोसेसिविटी ऑफ डीएनए पॉलीमरेज 1 रफली 3 to 200. 1 polymerase 1 polymerase 2 sorry this is 2 and this is 3. सो पॉलीमरेज 1 की जो प्रोसेसिविटी है कि एक एंजाइम कितने न्यूक्लियोटाइड को ऐड कर सकता है, उसके बाद उसकी एफिशिएंसी खत्म. सो इट कैन ऐड 3 to 200 नाइट्रोजनस न्यूक्लियोटाइड्स, नाइट्रोजनस बेसिस. पॉलीमरेज 2 कैन ऐड रफली 1500 न्यूक्लियोटाइड्स एंड पॉलीमरेज 3 कैन ऐड मोर देन 5 लाख बेस पेयर्स न्यूक्लियोटाइड्स. सो ये प्रोसेसिविटी 1 की है, ये 2 की है, ये 3 की है. सिमिलरली फिडेलिटी जो मैं बात कर रहा हूं कि किस स्पीड से कर सकता है, तो ये 16 से लेके 20 न्यूक्लियोटाइड पर सेकेंड. दिस कैन ऐड अप टू 40 न्यूक्लियोटाइड्स पर सेकेंड एंड दिस कैन ऐड रफली 252,000 न्यूक्लियोटाइड पर सेकेंड. तो ये था कंपैरिजन. ये दोनों का सबके पास होता है, सभी की प्रोसेसिबिलिटी होती है, सभी की फिडेलिटी होती है, किसी का ज्यादा होता है, किसी का कम होता है. तो जब मुझे लंबा डीएनए बनाना होगा, मान लीजिए मेरे पास एक 5 लाख बेस पेयर का डीएनए फ्रैगमेंट है जिसको मुझे रेप्लिकेट करना है. अगर मैं पॉलीमरेज 1 यूज करूंगा तो मुझे कितने नंबर ऑफ पॉलीमरेज 1 चाहिए होगा? क्योंकि 200 बेस पेयर अगर ये ऐड करता है एक एंजाइम, तो 1000 बेस पेयर ऐड करने के लिए 5 नंबर चाहिए. अगर मेरे पास 5 लाख लंबा बेस पेयर है तो मुझे कितने चाहिए? 100 से ज्यादा एंजाइम चाहिए होगा. 100 एंजाइम बनाने के लिए इतनी प्रोटीन सिंथेसिस बहुत एनर्जी इफिशिएंट प्रोसेस है, बहुत सारा एनर्जी खर्चा होता है उसमें प्रोटीन बनाने के लिए, और उतना स्पेस भी चाहिए. जबकि ये एंजाइम अगर आपको यूज करना है तो आपका एक एंजाइम ही सफिशिएंट है. तो whenever we want to replicate a larger piece of DNA, that time normally we go for DNA polymerase 3. जब मुझे छोटे छोटे टुकड़े के डीएनए बनाने होंगे तब मैं करूंगा डीएनए पॉलीमरेज 1, कि बड़ा बैटरी को मैं छोटे काम को लिए नहीं लगाऊंगा, छोटे एंजाइम को छोटे काम के लिए लगाऊंगा. एंड प्रूफ रीडिंग हम बोलते हैं जब किसी एंजाइम के पास एक प्रोसेस होता है जिससे वो अपनी गलती को सुधार ले. पॉलीमरेज का काम होता है न्यूक्लियोटाइड ऐड करना, अगर उसने गलत न्यूक्लियोटाइड ऐड किया उसको सुधार ले तो उसको बोलते हैं प्रूफ रीडिंग. ये हम आपको आगे बताते हैं किस एंजाइम के पास होता है, किस एंजाइम के पास नहीं होता और क्यों नहीं होता है. ठीक है? ये
तीनों में इस मतलब प्रूफ रीडिंग को लेकर के तीनों में कुछ डिफरेंस है क्या?
हाँ है ना. सब एंजाइम के ऊपर नहीं होना चाहिए, सब एंजाइम को जरूरत भी नहीं है. प्रूफ रीडिंग सभी एंजाइम को जरूरत भी नहीं होते हैं. प्रोकैरियोट में तो तीनों के पास होते हैं, 1 2 3 तीनों के पास होते हैं. यूकैरियोट में हमारे पास 5 एंजाइम होते हैं, 5 तरह का डीएनए पॉलीमरेज. तो सबके पास नहीं होता है, कुछ के पास होता है, कुछ के पास नहीं होता है. वो मैं आपको आगे बताता हूँ डिटेल. इन प्रोकैरियोट्स तीनों के पास.
इसमें एक्यूरेसी किसका ज्यादा रहता है? 1 का, 2 का कि 3 का?
नहीं, एक्यूरेसी सभी की बराबर होती है। ये ज्यादा न्यूक्लियोटाइड ऐड करेगा पॉलीमरेज 3, तो ऑब्वियसली इसकी एक्यूरेसी कम होगी। जो ज्यादा चलेगा ज्यादा गलती करेगा, जो कम चलेगा कम गलती करेगा।
ओके सर।
ये 5 लाख बेस पेयर ऐड करता है, तो गलती होने की पॉसिबिलिटी इसमें ज्यादा है। बट जब हम गलती करते हैं ना, तो हम 3 जगह पे गलती सुधारते हैं। इन केस ऑफ डीएनए पॉलीमरेज वी आर गोइंग टू ऐड द राइट न्यूक्लियोटाइड। उसके लेवल 3 लेवल्स हैं, 3 जगह हम चेक करते हैं। वो तीनों लेवल मैं आगे बताता हूँ आपको कहाँ कहाँ चेक करते हैं। पहला, अगर आपने गलती कर लिया, डीएनए पॉलीमरेज के पास एक पावर होता है कि वो सही न्यूक्लियोटाइड ऐड करे, वहां कुछ गलती सुधार लेते हैं कि हम सही न्यूक्लियोटाइड ही सेलेक्ट करते हैं। दूसरी गलती सुधारते हैं कि अगर हमने गलती कर भी लिया तो इस प्रूफ रीडिंग से हम गलती सुधार लेते हैं, तो ये भी आपको सही न्यूक्लियोटाइड देता है। और तीसरा होता है डीएनए रिपेयर सिस्टम। यहां भी गलती नहीं सुधारे तो डीएनए रिपेयर में जाकर सुधार लेते हैं। तो तीनों मिला के हमारे पास जो रफली होते हैं ना, 10⁹9 न्यूक्लियोटाइड ऐड करने पे एक गलती होती है। रफली इस तरह की एक्यूरेसी हमारे पास होती है कि इतना न्यूक्लियोटाइड हम ऐड करेंगे 10⁹9, तब एक गलती न्यूक्लियोटाइड होगा। ये प्रोबेबिलिटी है।
ओके सर, थैंक यू।
ये बहुत कम गलती है ना, मतलब 10⁹9 कितना हुआ, 100 करोड़ से ज्यादा हुआ। 100 करोड़ से ज्यादा न्यूक्लियोटाइड ऐड करेंगे और एक गलती करेंगे, बहुत माइनर गलती होती है ये, नॉर्मली वो भी नहीं होता है। ठीक है। रफली हमारा रेप्लिकेशन ये जो है ना, जो आपको दिख रहे हैं, ये दिस इज नॉट द कंप्लीट रेप्लिकेशन बबल, इट इज ओनली वन रेप्लिकेशन फॉर्क, हाफ ऑफ द रेप्लिकेशन फॉर्क। मतलब इस बबल का मैं हाफ पोर्शन देख रहा हूं यहाँ। तो जब हम रेप्लिकेशन बबल को देखते हैं, अब जैसे यहाँ से हम यहाँ हेलिकेज एंजाइम यहाँ आएगा, हेलिकेज एंजाइम विल कम हियर। इनिशिएट कैसे होगा वो मैं आगे आता हूँ, बताता हूँ आपको। हेलिकेज एंजाइम्स ब्रेक्स द हाइड्रोजन बॉन्ड, so both DNA strands will be unwinded. This will be unwinded and this will be unwinded. So here DNA unwinding is going moving in this direction और सुपर coiling होते जाते हैं। उसको हटाने के लिए हमारे पास गायरेज होता है, टोपोइसोमरेज होता है। अब आपको पता है डीएनए रेप्लिकेशन जब भी होगा, डीएनए न्यू न्यूक्लियोटाइड विल बी एडेड एट 3' OH एंड ओनली। जब भी नया न्यूक्लियोटाइड ऐड करेंगे वो OH पोर्शन पे भी ऐड होगा। सो आपके पास एक डीएनए फ्रैगमेंट होता है जहां 5' एंड पे क्या होता है, ये डीएनए का टुकड़ा है, इसमें 5' एंड पे क्या होगा, फॉस्फेट। एंड 3' एंड पे क्या होगा, OH। सो हमेशा जब भी व्हेनेवर वी आर गोइंग टू ऐड न्यू न्यूक्लियोटाइड विल बी एडेड हियर ओनली। सो डीएनए की डायरेक्शन हमेशा यही होती है, 5' से 3' की तरफ, बिकॉज नया न्यूक्लियोटाइड यही ऐड होगा। ठीक है। अब इस केस में जैसे हम देखते हैं ये कि ये हमारा डीएनए था ब्लू कलर का ओरिजिनल डीएनए, जिसका एक टुकड़ा ये है 3' और इसका दूसरा टुकड़ा ये है 5'। दिस इज सेकंड स्ट्रैंड जिसका 5' ये है, 3' ये है। आपको पता है कि डीएनए का जो स्ट्रैंड होता है वो एंटी पैरेलल होता है। तो ये अगर 3' डीएनए का टुकड़ा है तो जो कॉम्प्लीमेंट्री स्ट्रैंड होगा उसका ये 5' टुकड़ा होगा, बिकॉज इट शुड बी अपोजिट स्ट्रैंड्स। जो हमने देखा था उस समय में कि डीएनए स्ट्रैंड्स का अकॉर्डिंग टू वाटसन क्रिक मॉडल इट शुड बी एंटी पैरेलल, 3', 5', 3', 5', राइट। So if this is 3', so this should be 5'.
yes sir.
अब ये 5' है तो न्यू न्यूक्लियोटाइड इस डायरेक्शन में मूव करेगा, इधर ऐड होते जाएगा, करेक्ट। तो जैसे जैसे आपका डीएनए अनविंड होता जाता है, ये इस डायरेक्शन, ये डीएनए अभी तक यहाँ अनविंडेड है, थोड़ा और अनविंड होगा, ये पॉलीमरेज हमेशा ऐसे ऐड करते जाता है कंटिन्यूअसली। सो एक डीएनए स्ट्रैंड्स पे, टेम्पलेट पे कंटिन्यूअसली डीएनए इज ग्रोइंग। जैसे जैसे आपका डीएनए अनविंड होता जाएगा इसका लेंथ बढ़ता जाता है कंटिन्यूअसली। जबकि इस दूसरे टुकड़े पे देखिए ये 5' एंड है, तो इसका डायरेक्शन इधर होगा। अभी तक आपका डीएनए यहां तक अनविंड है, तो यहां तक डीएनए बन गया, यहां से शुरू हुआ यहां तक बन गया। अब आपने इस डीएनए को यहां से लेकर यहां तक अनविंड किया, तो फिर एक टुकड़ा यहां भी बनेगा, छोटा सा टुकड़ा। तो नॉर्मली जब रेप्लिकेशन होता है ना, तो एक स्ट्रैंड्स पे डीएनए कंटिन्यूअसली बनता जाता है, फ्रैगमेंट में नहीं होता है। तो यहाँ नॉर्मली डीएनए पॉलीमरेज 3 आता है, जो कंटिन्यूअसली इस डीएनए को बनाता जाता है, बिकॉज ये लंबा टुकड़ा बनना है। दूसरे स्ट्रैंड्स पे जो होता है ना, नॉर्मली छोटा छोटा टुकड़ा बनता है, ये जहां डीएनए पॉलीमरेज 1 नॉर्मली काम करता है, और इस छोटे टुकड़े को बोलते हैं ओकाजाकी फ्रैगमेंट। दिस इज कॉल्ड एज लॉन्गर डीएनए कंटिन्यूअस स्ट्रैंड्स, इसको बोलते हैं नॉर्मली लीडिंग स्ट्रैंड्स। बड़ा वाला टुकड़ा को बोलते हैं लीडिंग, एंड छोटे छोटे पीस जहां बनते हैं उसको बोलते हैं लैगिंग स्ट्रैंड्स। हमेशा कुछ रूल्स हैं, जैसे रूल्स हैं कि लीडिंग स्ट्रैंड्स जो होगा वो हमेशा उसी डायरेक्शन में मूव करेगा रेप्लिकेशन फॉर्क के तरफ। फॉर्क का एंड ये है, तो हमेशा ये फॉर्क के तरफ जाएगा लीडिंग स्ट्रैंड्स, एंड लैगिंग स्ट्रैंड्स जो होगा वो अवे फ्रॉम फॉर्क जाएगा। ये फॉर्क है तो इससे दूर जाएगा। आप यहां भी देखेंगे, इसमें भी अगर मुझे लीडिंग स्ट्रैंड्स बनाना होगा तो ये लीडिंग स्ट्रैंड्स बनेगा और यहाँ लैगिंग स्ट्रैंड्स बनेगा। ठीक है, लीडिंग स्ट्रैंड्स हमेशा फॉर्क के तरफ मूव करता है, लैगिंग स्ट्रैंड्स हमेशा फॉर्क से अवे मूव करता है। वो हम देखते हैं आगे डिटेल में। इस छोटे टुकड़े को जो डिस्कवर किया था ना, उनका नाम था ओकाजाकी, रफली अराउंड 1960s के अराउंड में, और उनके नाम पे ही बेसिस पे इनको इसको बोलते हैं ओकाजाकी फ्रैगमेंट्स। रफली 100 से 1000 बेस पेयर तक होता है, डिपेंडिंग ऑन प्रोकैरियोट्स एंड यूकैरियोट्स। हमारे में थोड़ा ज़्यादा छोटा होता है, प्रोकैरियोट्स में थोड़ा ज़्यादा बड़ा होता है। तो दिस इज कॉल्ड एज ओकाजाकी फ्रैगमेंट्स। तो ये नॉर्मल प्रोसेस है हाउ डीएनए रेप्लिकेशन स्टार्ट्स एंड हाउ इट इज गोइंग on continuously. Same picture, little bit more complicated. So this is your helicase enzymes which breaks these hydrogen bonds and DNA is going to unwinded. यहाँ DNA polymerase continuously bind होता जाता है इस डायरेक्शन में, this is called as leading strands. यहाँ छोटे छोटे टुकड़े में बनता जाता है, उसको बोलते हैं lagging strands. ठीक है, ये बेसिक सिंपल प्रोसेस हुआ। अब हम आगे देखते हैं how in details from where replication initiates and how its actual process is going on. इतना समझ में आ गया?
Yes, sir.
Any question, anything you have not understand? किसी के पास कोई क्वेश्चन है तो बताइए। No one is having any question. चलिए, पहले हम सिंपल में छोटे 6 स्टेप्स 7 स्टेप्स हैं, वो छोटे में हम देख लेते हैं, देन वी विल गो इन डिटेल्स। द फर्स्ट स्टेप अगर देखेंगे तो वी आर गोइंग टू हैव ए एंजाइम दैट इज कॉल्ड एज डीएनए हेलिकेज व्हिच ब्रेक्स द हाइड्रोजन बॉन्ड बिटवीन द बेसेस ऑफ द 2 एंटी पैरेलल स्ट्रैंड्स। लाइक ये आपका डीएनए है, तो यहाँ एक हेलिकेज एंजाइम होगा जो आपके हाइड्रोजन बॉन्ड को ब्रेक करेगा सो दैट बोथ स्ट्रैंड्स विल बी सेपरेटेड एंड मेक ए रेप्लिकेशन फॉर्क। अब जहां से शुरू होता है दैट पोर्शन इज कॉल्ड एज ओरिजिन ऑफ रेप्लिकेशन, ओरिसी साइट, एंड दे आर नॉर्मली एटी रिच रीजन, एडेनिन थाइमिन रिच रीजन। व्हाई एडेनिन थाइमिन रिच रीजन? एनीवन कैन गेस?
सर हाइड्रोजन बॉन्ड इजीली ब्रेक हो सकता है।
क्यों?
सर क्योंकि यहाँ पे डबल बॉन्ड्स होते हैं और जीसी रिच रीजन में ट्रिपल बॉन्ड्स होंगे।
यस, वो वहाँ जीसी अगर ज्यादा होगा तो 3 बॉन्ड्स होंगे, एंड एटी होगा तो 2 बॉन्ड्स होंगे। तो 2 बॉन्ड्स को ब्रेक करना थोड़ा इजी है, इसीलिए ओरिजिन ऑफ रेप्लिकेशन जहां होता है वहां एटी ज्यादा होते हैं, वहीं से हम इजीली आसान होता है तोड़ना, इसीलिए एटी रिच रीजन को हम ब्रेक करते हैं। बट देयर आर नंबर ऑफ प्रोसेसेस जो हम आपको डिटेल बताएंगे कल। तो ये है हेलिकेज रेप्लिकेशन फॉर्क इनिशिएट करता है, एंड देन डीएनए गायरेज या टोपोआइसोमरेज सुपर क्वाइलिंग को हटाता है। सुपर क्वाइलिंग अगर आपने घर में देखा होगा, मैं लास्ट में आपको एक मतलब अपना कैमरा ऑन करके दिखाता हूँ, एक सेकेंड। अगर मेरा कैमरा दिख रहा होगा आपको, इज इट विजिबल? ये जैसे दिस इज माय वायर विच इज क्वाइल्ड। अब मैं यहाँ से अनविंड करते जा रहा हूँ, तो इफ यूल सी हियर सुपर क्वाइलिंग हैज बीन इंक्रीज्ड। इनिशियली दिस वाज लिटिल बिट लेस, सुपर क्वाइलिंग वाज देयर। जब हम यहाँ से अनविंड करेंगे तो फर्दर में सुपर क्वाइलिंग बढ़ता जा रहा है यहाँ इस प्रोसेस में, पार्ट में। सो इस सुपर क्वाइलिंग को हटाने के लिए हमारे पास टोपोइसोमेरेज होता है। हेलिकेज वर्क्स हियर, सो इट मूव्स इन दिस डायरेक्शन लाइक दिस वन, और फर्दर में सुपर क्वाइलिंग बढ़ता जाता है, जिसको हटाने के लिए हमारे पास टोपोइसोमेरेज और गायरेज होते हैं। So in step first, we are going to get this type of a structure which is called as replication fork. अब जब हमने इसको ओपन कर दिया तो ये सिंगल स्ट्रैंडेड डीएनए हो गया, विच इज लिटिल बिट मोर फ्रेजाइल। यहाँ एंजाइम्स काम ब्रेक कर सकता है इसको, तो यहाँ स्टेबलाइज करने के लिए छोटे छोटे प्रोटीन्स बाइंड करते हैं, जिसको बोलते हैं एसएसबी प्रोटीन, सिंगल स्ट्रैंडेड बाइंडिंग प्रोटीन या सिंगल डीएनए स्टेबलाइजिंग प्रोटीन, जो आपके सिंगल स्ट्रैंडेड डीएनए को थोड़े स्टेबल करते हैं, एंजाइम की एक्टिविटी नहीं होने देते हैं। तो दिस इज माय बेसिक्स ऑफ स्टेप नंबर 1। इन स्टेप नंबर 2, अब ये टुकड़ा यहाँ है, जब आपके पास मान लीजिए ये डीएनए स्ट्रैंड था, यहाँ कुछ भी नहीं था, तो मैंने क्या बोला था, डीएनए पॉलीमरेज कैन नॉट स्टार्ट फ्रॉम द जीरो। तो यहाँ डीएनए पॉलीमरेज शुरू नहीं कर सकता, तो एक छोटा सा टुकड़ा पहले चाहिए यहाँ। वो कौन बनाएगा? दैट इज ए प्राइमेज एंजाइम विच इज केमिकली आरएनए पॉलीमरेज। वो बनाता है छोटा सा टुकड़ा, जिसको बोलते हैं आरएनए प्राइमर। सो in both leading strands also and lagging strands also, we are going to initiate RNA synthesis by a primase enzyme, that is chemically RNA polymerase, which makes a small fragment of oligonucleotide, that is called as RNA primer, और ये प्लेटफॉर्म प्रोवाइड करता है डीएनए पॉलीमरेज के लिए। अब जब यहाँ टुकड़ा बन गया, तो नाउ हियर वी आर हैविंग OH group. So DNA polymerase will come and add a fresh nucleotide here, and then it will slightly enlarge. This provides the starting point for DNA replication. This is step number 2. Then step number 3, on one strands, a replication is continuously going on towards the fork, that is called as leading strands. On another strands, it is in fragments, that is called as lagging strands. बट डायरेक्शन दोनों की 5 प्राइम टू 3 प्राइम ही होगी। सो ऑन वन स्ट्रैंड्स लीडिंग स्ट्रैंड्स, ऑन अनदर स्ट्रैंड्स वी आर हैविंग लैगिंग स्ट्रैंड्स और ओकाजाकी फ्रैगमेंट्स। जब ये व्हेन वी हैव दिस मच, देन वी आर गोइंग टू रिमूव दिस प्राइमर पोर्शन यहां से, और दीज प्राइमर्स नीड टू बी रिमूव्ड। तब वहां हमें फिर से चाहिए होता डीएनए पॉलीमरेज 1, जो छोटे छोटे फ्रैगमेंट्स को हटाएगा, ओकाजाकी फ्रैगमेंट्स को ज्वाइन करेगा, पॉलीमरेज को, प्राइमर को हटाएगा, सो दैट कंप्लीट डीएनए। इनिशियली क्या था, आप यहां देखेंगे तो इसमें आपका डीएनए का पोर्शन ये है, दिस इज डीएनए, दिस इज आरएनए, दिस इज डीएनए, दिस इज आरएनए, दिस इज डीएनए, दिस इज आरएनए। सो वी हैव डीएनए आरएनए मिक्स्ड। ऐसे नहीं, मतलब ऐसा नहीं हो सकता। सो वी नीड टू रिमूव दिस आरएनए पोर्शन। So RNA portion need to be removed by DNA polymerase 1, and उसके जगह पे नया DNA बनेगा। So primer will be replaced by a DNA fragment by DNA polymerase 1, and later on those portions will be joined by DNA ligase. तो ये कंप्लीट हमारा फ्रैगमेंट बन जाता है लैगिंग स्ट्रैंड्स का भी लास्टली इस तरह से। तो दीज आर द 7 स्टेप्स जिसको हम डिटेल में देखेंगे टुमारो। ठीक है, ये बेसिक्स था कि हाउ वी स्टार्ट फ्रॉम स्टेप 1 एंड देन हाउ इट एंड्स विद द ligation. So we will see details about the DNA polymerase, differences between the DNA polymerase, eukaryotic DNA polymerase, prokaryotic DNA polymerase, and then how it initiates and how it is further goes on. If possible, tomorrow I will show you a small video clip also, cartoons also, so आपको थोड़ा ज़्यादा समझ में आएगा. If और कोई किसी का क्वेश्चन है इसमें से पूछिए. If you have any question, you can ask.
सर एक क्वेश्चन था.
हाँ हाँ बताइए.
कि जो ओकाजाकी फ्रेगमेंट है सिर्फ लैगिंग स्ट्रैंड में क्यों बनता है, लीडिंग स्ट्रैंड में क्यों नहीं बनता है?
क्योंकि वहां अगर आप ये स्ट्रैंड्स देखेंगे ना, जैसे ये हमारा डीएनए था, ठीक है. अब ये है हमारा 3 प्राइम एंड, ये है 5 प्राइम एंड, ये है 5 प्राइम एंड, ये है 3 प्राइम एंड. अभी डीएनए यहां तक अनविंड हुआ है, ठीक है. तो यहां से हमने बनाना शुरू किया, तो ये बनता जाएगा, यहां तक बन के आया, ये हुआ 5 प्राइम एंड और ये हुआ 3 प्राइम एंड. अब आपने डीएनए को थोड़ा और आगे तक अनविंड कर लिया, यहां तक अनविंड कर लिया, तो ये तो इसी डायरेक्शन में बनता चला जाएगा, करेक्ट. तो ये लीडिंग स्ट्रैंड्स कंटिन्यूअसली गोज ऑन एज डीएनए विल बी अनविंड. जैसे जैसे आप डीएनए को ओपन करते जाएंगे, ये आगे आगे बनता जाएगा. अब दूसरे स्ट्रैंड की मैं बात करता हूं. मान लीजिए डीएनए हमने यहां तक अनविंड किया है, तो यहां का डायरेक्शन है 3 प्राइम, यहां था 5 प्राइम, तो यहां डायरेक्शन ये होगा बनने का. तो अभी आपने यहां तक बना ओपन किया हुआ था, यहां से तो बना लिया हमने. अब आपने डीएनए को अनविंड कर दिया यहां तक, तो इस पोर्शन पे कैसे बनेगा, नया टुकड़ा बनना चाहिए. इस स्ट्रैंड पे तो ये कंटिन्यूअसली जाता जा रहा है, बिकॉज इसकी डायरेक्शन है 5 to 3. दिस इज 5 प्राइम, दिस इज 3 प्राइम, सो इट इज कंटिन्यूअसली गोइंग ऑन. यहां का डायरेक्शन 5 3 होना है, तो 5 इधर आएगा, 3 इधर आएगा, तो ये तो उल्टा डायरेक्शन में होगा ना, ये तो अवे फ्रॉम फॉर्क होगा. तो एक स्ट्रैंड पे जैसे जैसे डीएनए अनविंड होता जा रहा है, ये बनता जाता है आगे तक, क्योंकि उसी सेम डायरेक्शन है, 5 3 मेंटेन कर रहा है. दूसरे पे 5 3 मेंटेन करने के लिए छोटे छोटे टुकड़े में बनाना पड़ता है, बिकॉज डीएनए धीरे धीरे खुलता है, एक ही बार में पूरा नहीं खुल जाता है. तो इसीलिए जैसे जैसे डीएनए खुलता जाएगा, छोटा छोटा फ्रैगमेंट हम बनाते हैं दूसरे स्ट्रैंड्स पे. पहले जैसे अगर आपने ये पिक्चर देखेंगे, पहले मान लीजिए डीएनए हमने यहीं तक अनविंड किया था, इस पोर्शन तक, तो ये टुकड़ा बना. फिर आपने यहां तक अनविंड किया, तो ये तो कंटिन्यूअसली बनता जा रहा है इस डायरेक्शन में, बट यहाँ पहले आपने ये टुकड़ा बना लिया, फिर थोड़ा और डीएनए ओपन किया तो ये टुकड़ा बनाया, फिर और थोड़ा अनविंड किया तो ये टुकड़ा बनाया. आपने यहाँ तक अनविंड करेंगे तो ये तो कंटिन्यूअसली बनता जाएगा, जो आपको यहाँ से बनता आ रहा है, बट यहाँ पे आपको फिर से एक टुकड़ा बनाना पड़ेगा. तो इसीलिए एक पे कंटिन्यूअसली बनता जाता है, जिसको हम लीडिंग बोलते हैं, दूसरे पे छोटे छोटे फ्रैगमेंट में बनते जा रहे हैं, जिसको हम लैगिंग बोलते हैं. जस्ट टू मेंटेन द डायरेक्शन ऑफ 5 प्राइम to 3 प्राइम डायरेक्शन, इस डायरेक्शन को मेंटेन करने के लिए.
ओके, थैंक यू सर.
एनीवन एल्स? और किसी के पास कोई क्वेश्चन है तो पूछिए. नो वन इज हैविंग एनी क्वेश्चन? आज कम स्टूडेंट्स क्यों हैं? या तो समझने में दिक्कत हो रही है?
नो सर.
तो कम स्टूडेंट्स हैं आपके?
नो सर, सही है.
आज तो स्टूडेंट्स कम हैं आपके. अगर आपको दिक्कत हो रहा था आप बताएंगे, तभी मैं स्लो हो जाऊंगा या और धीरे पढ़ाऊंगा. धीरे पढ़ाना चाहिए, राइट? यस और नो कुछ तो बोलिए.
यस सर, एवरीथिंग इज फाइन.
आप एक बार आपस में ग्रुप में डिसाइड बात कर लीजिएगा. अगर किन्हीं को कोई दिक्कत है ना, अगर मैं थोड़ा ज़्यादा फास्ट हूँ या समझने में दिक्कत हो रही है तो मुझे बताएंगे, देन आई विल बी लिटिल बिट मोर स्लोअर. ओके
ओके सर.
ठीक है। आप क्या एक चीज, दो चीज मैं बुझा, अगर मैं स्लाइड्स आपको कैसे दूं, वो आप मुझे बात करके मैसेज कर दीजिए उस पर, टेलीग्राम पे अगर आपने ज्वाइन किया है तो किसी का मेल आईडी मुझे भेज दीजिएगा, मैसेज कर दीजिए, तो मैं उनको स्लाइड्स भेज दूंगा। ठीक है, एक वो चीज़। दूसरा, कुछ वीडियोज होंगे जो मैंने एक यूट्यूब चैनल बनाया है। ठीक है, अगर आप में से किसी ने विजिट करा होगा तो मेरा यूट्यूब चैनल है इस नाम से, क्लिनिकल बायोकेमिस्ट्री बाय पीके प्रभाकर के नाम से। अब वहाँ जाएंगे, कुछ वीडियो आपको डीएनए रेप्लिकेशन सब पे छोटे छोटे टुकड़े में मिलेंगे वहाँ भी। ठीक है,
यस सर।
सर चैनल कौन सा है?
ये क्लिनिकल बायोकेमिस्ट्री बाय डॉक्टर पीके प्रभाकर के नाम से सर्च करेंगे ना तो मिल जाएगा आपको। अदरवाइज मैं चैट बॉक्स में आपको एक बार डाल देता हूँ।